polysaccharides are the most abundant biopolymer available in nature. This group of molecules can be found in microorganisms, plants, and animals. They perform different functional roles in living beings. Most of the polysaccharides are present in plants and vegetables, which consequently represent the major source of these biopolymers and can be exploited for a variety of purposes. Indeed, polysaccharides have found applications in almost all sectors of human life. These biopolymers are used in food industries, pharmaceutical industries, biomedical industries, and several other biotechnological applications, including production of biofuels. Starch, pectin, cellulose, and hemicelluloses are the most studied and extracted plant polysaccharides. Although various types of polysaccharides are extracted from the vast plant-based sources, all these are associated with both economic and environmental cost. This does not only depend upon the techniques used for extraction of the polysaccharides, but also affected by the availability of the feedstock. Hence, the search of alternative sources for polysaccharides is crucial to ensure round-the-year availability at a reasonable price without affecting the environment and the food chain. A variety of organisms produce exopolysaccharide as an extracellular or cell surface attached material in the form of amorphous slime. These polysaccharides can also be classified as homopolysaccharides and heteropolysaccharides. Most bacteria are used as industrial producers of exopolysaccharides. Xanthan, gelen, curdlan, and dextrin are the well-known industrial produced polysaccharides, and they are synthesized by the species of Xanthomonas, Spingomonas, Alcaligenus, and Leconostac. Few fungal exopolysaccharides are also well known for their unique physico-chemical properties and potential for wide industrial applications. Pullulan is one such commercially important fungal exopolysaccharide. This is a water-soluble alpha-glucan composed of maltotriose subunit. This extremely versatile exopolysaccharide has the capability of providing a technology platform for product innovation. Due to several unique physico-chemical properties, Pullulan has found wide application in food, pharmaceutical, and cosmetic industries. Recent literature suggested its potential towards application in the field of drug and gene delivery. It is an excellent film former, producing a film which is heat sealable with good oxygen barrier properties and which can also be printed. Hence, Pullulan can be formed into capsules for use with pharmaceutical and nutraceutical products. The global market size of Pullulan was 10,000 TPA, that is $250 million in 2009. It was expected to grow at 6.75% per annum. More important, its major competitive products, that is gelatin and xanthan gum, also have cumulative market size of more than $2 billion, which can also be tapped to enhance the total market of Pullulan. The major manufacturer of this molecule are Hayashibara, NOF Corporation, and Yushi Seihin Co. Limited, all from Japan.
Despite large number of uses, there are certain problems associated with Pullulan production. One of them is the producing organism has a tendency to produce melanin, which actually enhances the recovery and purification cost. Ultimately, the major bottleneck for commercial production of Pullulan comes down to the fact of the cost of the final product. And the final product cost is 3 times more as compared to its competitive products like xanthan, gelen, etc. So we wanted to address this challenge by screening different type of strains, and our objective was initially to start with to obtain highly osmotolerant, high exopolysaccharide producing strains. And I collaborated with my colleague Dr. G.S. Prasad, head MTCC, and we have started with the objective to obtain a technology for economically feasible production of Pullulan.
As best of our knowledge, at present, there is no industrial manufacturer of this molecule in India.
Our research group works on the diversity of yeast from various ecological niches. One of them is to study the association of yeasts and flowers. So the actual work related with Pullulan is an offshoot of the yeast biodiversity work, in which actually we are looking for yeasts which are osmotolerant and non-pigmented. We have isolated several yeasts as part of our project and found that some of them belong to the genus Arabidopsis pullulans. When we have screened them for the production of Pullulan, we found about 10 of them producing very good amounts of Pullulan.
During the process, the initial optimization is carried out in shake flask and further transferred to laboratory scale fermenters. In the beginning, we screen all Arabidopsis strains for Pullulan production using a media containing glucose, yeast extract and peptone. Each strain was grown for 144 hours and the Pullulan was precipitated from cell-free fermentation broth using cold ethanol. 5 of the highest Pullulan production strains were considered for second stage screening depending on their ability to produce exopolysaccharide. Among these, the strain designated as Arabidopsis pullulans RBF4A3 was found to be the most efficient Pullulan producer. A techno-economic calculation indicated that media components used were adding significant cost to the production. Keeping this in mind, the potential of agri-industrial residue as nutrient in place of conventional media components like yeast extract and peptone for Pullulan production was explored. Analysis of raw production cost component for Pullulan production suggested that sole utilization of agri-residues led to the cost reduction of raw material by more than 90%.
In a nutshell, in CSIR IMTech, we have developed a technology for production of Pullulan by using an osmotolerant non-pigmented strain of Arabidopsis pullulans. The process has been initially optimized in shake flask level and further transferred to laboratory scale fermenter level and now being taken up to higher scales of 100 liter and 500 liter fermenters. In this process, we are able to produce more than 70 gram per liter Pullulan production, which is higher as compared to most of the literature reports, including publication as well as patents. More importantly, our process talks about production of Pullulan by using agri-industrial waste. Actually, we are calling this process for creating wealth from waste, which are actually very low cost raw material. And our process is totally indigenous. The organism is from India and the whole process has been designed and created in India, so it goes very well along with the mandate of Government of India, which says make in India. So we are trying to produce Pullulan by indigenous Indian technology.